Hi all, today we are going to discuss about photosynthesis in higher plants. So we will have a short discussion with maximum questions from this area. So in the theory itself I will tell how all the questions can be coming from this topic. So let me start with photosynthesis in higher plants. And maximum expected question from this chapter is 3. Minimum 1 or 2 questions will be surely coming for the NEET exam. So you don't have to read the chapter a lot for learning this. You can have this capsule video for this. So let me start with photosynthesis in higher plants. So let me tell what is photosynthesis. It's the process by which green plants are preparing their own food using the raw materials like carbon dioxide and water in presence of sunlight and chlorophyll. And here they will be using 6 carbon and 12 water molecules in presence of sunlight and chlorophyll, and it will be getting converted into carbohydrate C6H12O6. So a hexose sugar is formed, and here they will be releasing 6 oxygen and 6 water molecule. So this is the general chemical equation for photosynthesis. Then understand photosynthesis is a anabolic process. So what is anabolic? Anabolic means constructive process, where you can see small substances are combining to form a complex molecule. So here the smaller particles that are taking part in photosynthesis is carbon dioxide and water. And the complex particle that is formed is carbohydrate, the hexose sugar, glucose. So here, first point you should remember, photosynthesis is a anabolic process. Anabolic means it's a constructive process. And second thing, it's a redox reaction. Photosynthesis is a redox reaction, where you can see the carbon dioxide is actually getting reduced. So that's a question. What is getting reduced in photosynthesis? The answer is carbon dioxide. And there is oxidation of water also. So water undergo oxidation, where you can see they will be releasing out the oxygen. So oxygen in photosynthesis is coming from water molecules. So there is breaking of water molecule, releasing the oxygen. The process is called photolysis. Now, remember, photosynthesis is an anabolic process and it's a redox reaction, where you can see the carbon dioxide is reduced and the water is undergoing oxidation. And here, in photosynthesis, it's also called carbon dioxide assimilation. The process is also called as carbon dioxide assimilation. So, where this photosynthesis is taking part in the plant, that will be our next discussion. So these are the general things about photosynthesis. It's mainly occurring in the plants, especially the green parts and all. It's an anabolic process or a constructive process, where you can see the carbon dioxide and water, it will be combining to form carbohydrate. These are smaller particles. They are forming a complex particle, that is carbohydrate. Then the second thing you should understand, it's a redox reaction, where you can see carbon dioxide is reduced into carbohydrate and the water undergo oxidation in order to release oxygen. So whatever I said is a brief description. Now we are going to elaborate this topic. So about the reduction, oxidation, how it is taking place, how the carbon get fixed, etc. we will start discussing. Before that, let's discuss the site of photosynthesis. Where is photosynthesis occurring in the plants? You know that in plants, the photosynthesis is mainly confined to the green parts of the plant. The photosynthesis is mainly taking place in the green parts of the plant. And in the green part itself, it's mainly taking place in the leaf. And in the leaf, where is the photosynthesis taking place? Answer, in the leaf, photosynthesis is mainly confined to mesophyll cells. You might have learned the anatomy of flowering plants, the cross section of leaf, dicot leaf and monocot leaf. So to get more idea, let's have a discussion about the anatomy of dicot leaf. And in the mesophyll, the main site of photosynthesis that we will be discussing is chloroplast. So let's have a discussion about the main site of photosynthesis. So in the mesophyll, photosynthesis is mainly confined to the chloroplast. So structure of a leaf, then we will discuss the structure of chloroplast. So first of all, we are going to discuss the structure of a dicot leaf. The dicot leaf structure that is given in NCERT is here, but I will just draw so you will get an idea how it is. So here in the plants, if I take, you can see there is petiole that is extending to form the midrib and all, and there is leaf. So this leaf cross section only I am going to draw. So this is the midrib that I will draw like this when I am drawing the cross section, that is also given in NCERT textbook. Hope you can understand. So in the leaf, you already know the petiole will be carrying this xylem and phloem. So in yellow color, I am drawing a xylem. This xylem is passing through the midrib of the leaf. Got it? From the petiole, it is extending into the midrib, and it also forms the veins as well as it's also found in the veinlets. And there is also phloem that is passing through the stem, and it's also entering into the leaf through petiole, and it is also extending through the midrib of the leaf. It's also actually coming in the veins as well as veinlets. So this leaf cross section I am going to show, and in that where is the photosynthesis occurring and all, we will have a small discussion. So cross section of the leaf, first of all let me draw the midrib of the leaf. The midrib of the leaf will be having upper xylem and the lower phloem. Hope you can understand. And it's surrounded by the bundle sheath. So bundle sheath is actually made up of parenchyma. Then there is upper epidermis on the dicot leaf. Then there is lower epidermis also. So on the upper epidermis, a thick cuticle will be present. So what I am drawing is a cross section of a dorsiventral leaf or dicot leaf. So dicot leaf, if you are taking, this leaf is actually perpendicular to the stem. More sunlight will be falling on the top. That is the reason they have a different anatomy or internal structure. So when more sunlight is falling on the top, the mesophyll cells are packed like that to capture more light energy. And to reduce more transpiration or reduce the rate of transpiration and loss of water, majority of stomata is located on the lower epidermis. So here there is upper epidermis and the lower epidermis. Now just look at this diagram. So here adaxial and abaxial epidermis, midrib I have drawn like this. Now cross section we will be drawing straight. Hope you have understood. Now in between the upper and lower epidermis, so upper epidermis is also called as adaxial epidermis. Then lower epidermis, that is actually called as abaxial epidermis. So adaxial epidermis, upper epidermis, or it is ventral in position. Don't think that is dorsal. Understand upper position, that is actually the ventral, and this one is the dorsal. So many questions are there even from anatomy, like statements. In dicot leaf, most of the stomata is located on the ventral surface. That is wrong. Ventral surface is adaxial. Most of the stomata is located on dorsal surface. Like that you have to actually learn all these. Upper epidermis, adaxial epidermis, or it's the ventral surface. Mostly the sunlight is falling on the ventral surface. And the dorsal surface, you can see that is the bottom, that is the lower epidermis. Pale green in color and all in the leaf you can see. Majority stomata in the dicot leaf is located on the lower epidermis, that is dorsal in position. And even the cuticle, if you are taking, more thick cuticle will be coming on the upper epidermis and very less thick cuticle will be coming on the lower epidermis. Then in between the upper and lower epidermis only the mesophyll cells are coming. So compactly packed mesophyll can be observed here. That compactly packed mesophyll that is coming here is called as palisade mesophyll. So like this is the palisade mesophyll that will be coming just below the adaxial epidermis. So like this, just below the adaxial epidermis or upper epidermis, you can see there is a compactly packed mesophyll that is coming. Then there is another mesophyll that is coming. So this mesophyll is called as palisade mesophyll. Then there is another mesophyll that is coming here, which will be having a lot of air spaces in between. That mesophyll is actually called spongy mesophyll. So like this, two types of mesophyll is present. So this is the palisade mesophyll and this is the spongy mesophyll. So spongy mesophyll is having a lot of intercellular air space. So why they have this anatomy? Then there is bundle sheath extension also that is coming from this vascular bundle. So from the vascular bundle you can see there is a lot of bundle sheath extensions that is coming. This bundle sheath extensions will be extending from the bundle sheath up to the upper epidermis as well as it will be extending up to the lower epidermis. So like that there is a bundle sheath extension that is coming here. So bundle sheath extensions will be having or it will be extending from upper epidermis to the bundle sheath and the lower epidermis to the bundle sheath. So this is the bundle sheath extension. This is made up of parenchyma. So this is the anatomy or the internal structure of a dicot leaf. So the leaves are well adapted for gaseous exchange. So all these structures will be helping them in a good gaseous exchange, especially for photosynthesis and respiration. So here, photosynthesis, the carbon dioxide should reach the site of photosynthesis. The main site of photosynthesis is what? Chloroplast. So I will be just putting some yellow dots representing the chloroplast. Understand, more number of chloroplast is located in the palisade mesophyll and it's also coming in the spongy mesophyll. So this yellow dots that I am putting, it is representing the chloroplast. You might have read one line in Cell, the basic unit of life chapter. Around 20 to 40 chloroplast is present in a mesophyll. That you should connect in this chapter also because questions are there from that area also. So mesophyll, the number of chloroplast that is coming in a mesophyll is 20 to 40 chloroplast per cell can be observed in the mesophyll. That is also a question area. And majority chloroplast will be located inside the palisade mesophyll. So don't think palisade mesophyll is having only the chloroplast. They will be having a large central vacuole and the peripheral nucleus and the chloroplast. All these things will be coming. So this yellow dots which I am putting, it represent the chloroplast. Understand, this chloroplast is located in the spongy mesophyll as well as it is located in the palisade mesophyll and it's also coming even in the guard cell. Guard cells also will be having the chloroplast. So the main site of photosynthesis in the green plants. If a question is coming, remember the main site of photosynthesis will be the green part of the plant. In the green part, it is mainly occurring in the leaf. In the leaf, it will be mainly occurring in the mesophyll. In the mesophyll, it is mainly occurring in the chloroplast. So now let's discuss the structure of a chloroplast. Before going there, how the raw materials are delivered to this chloroplast, let's have a discussion. So understand, from the xylem, you know that the water is entering into the root, into the xylem, and it will be going into the xylem upward. and it is reaching up to the midrib. Hope you got it. So like this, look at here, this yellow line represents the xylem. So water is entering from the root into the, from the soil into the root, mainly in the xylem. Then it will be going upward. Then it will be going through the petiole in the midrib, and in the veins and all they will be going. And from this xylem, the water will be delivered to all these mesophils. So understand, the xylem is having water, and the water will be mainly going to where all these mesophil cell, especially the chloroplast in the mesophil, will be getting water from the xylem, and using that only they are going to perform photosynthesis. Also understand, some water will be lost out through the stomata as water vapor. The process is called transpiration. So there is diffusion of water or water vapor from higher concentration inside the leaf to lower concentration in the atmospheric air. The process is called as transpiration. So all these cells, mainly the chloroplast in the cell, will be getting water from xylem, that is present in midrib of the leaf, veins of the leaf and veinlets of the leaf. Then carbon dioxide is also entering into the leaf. That is mainly from atmospheric air. It is diffusing and reaching all the mesophil cells, chloroplast. So this, an outline should be there in the mind. So what is photosynthesis? The process by which green plants prepare their own food. And here what is going on? It's an anabolic process, where you can see they are constructing the things using carbon dioxide and water. So carbon dioxide is reduced into carbohydrate, and where it is occurring, it's mainly occurring in the green parts of the plant. Especially it is confined to the leaf. In the leaf it is occurring in the mesophil. In the mesophil, the process take place in chloroplast. So chloroplast should get two things. So how the two things are coming? First thing is carbon dioxide, that is from atmospheric air. Hope you understood the internal structure of leaf and how this carbon is entering into each and every cells in the chloroplast and all, or in the chloroplast, how they are reaching, you should understand. Then water is supplied by the xylem. So hope you got an introduction part of this chapter. Now let's discuss about chloroplast and what is going on in the chloroplast. So if you take the chloroplast, you might have already learned the structure of chloroplast in the Cell, the Basic Unit of Life chapter. It's a double membrane bound structure. So if I take one of the mesophil, understand, in the periphery there is cytoplasm. At the center of a mesophil there is a vacuole. So in the peripheral cytoplasm, there can be around 20 to 40 chloroplasts. It's only one chloroplast and cup-shaped in Chlamydomonas. But here you should understand, in mesophil there are many chloroplasts, around 20 to 40 chloroplasts per cell. Now, chloroplast structure. It's double membrane bound. So this structure is given in NCERT. You have to refer the chloroplast diagram from Cell, the Basic Unit of Life, as well as you have to check the diagram in photosynthesis chapter. Anyway, a diagram I will be drawing so you can get more idea from the diagrams. So here chloroplast will be having two membrane. So there is an outer membrane and there is also an inner membrane. So there is a dual membrane structure that is coming in the chloroplast. There is an outer membrane and there is an inner membrane. So like this, two structures are coming in the chloroplast. And in between that membrane, so it's a double membrane bound structure. It's having outer membrane and inner membrane. In between this membrane, there is a space. That space is called periplasticidal space. And it's getting filled with a fluid that is called as periplasticidal fluid. So here, there is a periplasticidal space which is getting filled with periplasticidal fluid. Now, this inner part of the chloroplast, a fluid is coming. That part is called as stroma. In the stroma, they will be having a double-stranded circular DNA. And from this double-stranded circular DNA, they can actually produce what? They also have RNA. And they also have, from the RNA can be read by ribosome. they also have 70S ribosome. And they can also have some proteins or enzymes. So understand, it's like a cell. It's partially like a cell. They have their own DNA, RNA, ribosome, even enzymes and proteins are present in the stroma. And which type of ribosome is present in chloroplast? The answer is 70S. So generally when we ask the students what is the type of ribosome found in eukaryote, they will tell 80S only. Understand, eukaryote have both 80S and 70S. In the endoplasmic reticulum, rough endoplasmic reticulum and all, the type of ribosome that is found is 80S. But coming to cell organelles like chloroplast and mitochondria, the type of ribosome that is found is 70S. So please remember, the eukaryotic cells are having both 80S and 70S ribosome. Then it's a semi-autonomous cell organelle. Partially they can work on its own. And it's like a cell itself. Partially it's behaving like a cell itself. So it's a semi-autonomous cell organelle. One more question. Semi-autonomous cell organelles in eukaryote, mitochondria and chloroplast. They are also called endosymbionts. Now, they also have the most abundant enzyme protein in the biosphere. This is a connection with biomolecules chapter, that is rubisco. The most abundant enzyme protein in the biosphere is rubisco, that is found in the stroma of chloroplast. So one of the enzyme I am writing here, that is rubisco. So hope you got this much questions which I was telling. Photosynthesis is which type of reaction? It's an anabolic process. It's a redox reaction. What is reduced? What is oxidized? Where is it taking place? Anatomy of the dicot leaf. Where is the stomata located? All these are question areas. Then what is the number of chloroplast per mesophyll cell? That is also a question area. Then which is a semi-autonomous cell organelle, chloroplast and mitochondria. Why it is called semi-autonomous? Because it can function by its own, partially. They have their own DNA, RNA, protein, ribosome, etc. Which is the most abundant enzyme protein in the world. That is rubisco. Where is it located? It's found in the stroma of chloroplast. Now, they have some coin-like structures here. That coin-like structures in the stroma is called as thylakoids. And there is a stack of thylakoid that is coming. That stack of thylakoid that is coming is called as grana. Around 2 to 100 thylakoids are coming. That is actually called as grana. So remember, 2 to 100 thylakoids together is called grana. And these grana are interconnected by another structure. That structure which is going to connect this grana is called as stroma lamella. So that's all about the structure of a chloroplast. Stroma lamella is also called intergranum, Fred channels, etc. So in different question bank, when you are doing, you may be coming across all these. So understand the structure of chloroplast. It is double membrane. There is an outer membrane. There is an inner membrane. Space between that is called periplasticidal space. It's filled with periplasticidal fluid. And you can see there are coin-like structures coming here. That coin-like structures are called thylakoid. Thylakoids are stacking together in order to form grana. And they are interconnected by stroma lamella, which is also called as intergranum or Fred channels. Then we are going to divide the chapter into two, light reaction and dark reaction. So where is the light reaction taking place and where is the dark reaction taking place? Let's have a small discussion. So first of all, let me tell, light reaction will be occurring in the grana or the thylakoid because they will be having chlorophyll pigments. So pigments are located on this thylakoid. So here in the thylakoid only, the light reaction will be occurring. So our first discussion will be light reaction. So the light energy from the sun will be getting converted into two chemical energy that happens in the thylakoid. They harvest the light energy and using that they can produce two chemical energy. So radiant energy from the sun is getting converted into chemical energy. So they will be releasing out these two chemical energy in the stroma, that is ATP and NADPH. So NADPH plus H plus. So like this, two chemical energy will be produced in the grana using sunlight. This process is called as light reaction. Using that only, you can see the carbon dioxide will be fixed by the enzyme Rubisco in the stroma. So using this energy from light reaction, so light is not required, using energy from the light reaction, that is ATP and NADPH, in the stroma, this Rubisco enzyme can fix carbon dioxide into the hexose sugar. This process is the second part of the chapter, that is called as dark reaction. Hope you got it. Then one more question. Which is the third membrane system of chloroplast? Answer, thylakoid membrane. So I already have discussed two membrane, outer membrane and inner membrane. Other than that, one more membrane is coming in the chloroplast, that is thylakoid membrane. So thylakoid membrane is considered as the third membrane system of chloroplast. Hope you got this much. So a brief up of chapter is here. We are going to divide photosynthesis into two, light reaction and dark reaction. Light reaction occurs in the grana, where you can see the light energy is going to get converted into chemical energy like ATP and NADPH. That is, in the stroma side they are getting prepared. Using that ATP and NADPH, in the stroma, the Rubisco will be performing the dark reaction. They don't need light, but they need energy from the light reaction. That is why we call it as dark reaction. So they will reduce the carbon dioxide into carbohydrate. So this process is carbon dioxide assimilation. And the energy used in carbon dioxide assimilation is called as assimilatory power. Especially ATP and NADPH will be considered as assimilatory power. How they are produced and all will be our next discussion. Even in the light reaction, there is photolysis of water. So where it is taking place and all, I will be discussing later. Now just check a brief up. So in photosynthesis, first we are going to discuss about light reaction. It's also called photophosphorylation. So why it is called as photophosphorylation? Because here energy is produced using sunlight. So what all things are produced? One is ATP. Second is NADPH. That's also an energy. Then third, there is photolysis of water. Water will undergo breaking using sunlight, and here it will be resulting in the formation of, so photolysis of water can result in the release of oxygen and H plus. So this is photolysis of water. So this much we will be discussing in light reaction. So what I have written is a brief up only. Understand, light reaction will be occurring in the grana, where the light energy is converted into chemical energy like ATP and NADPH, and there is also photolysis of water. And using this chemical energy from the light reaction, there is dark reaction that will be taking place. So light reaction is mainly occurring in where? Grana. And second part of the chapter, we will be discussing about dark reaction. This dark reaction will be occurring in the stroma. And in the stroma, you can see by using the enzyme Rubisco and all, this carbon dioxide will get assimilated as the hexose sugar. So this process or this chemical energy is actually called as assimilatory powers. They are called as assimilatory powers because they are used for carbon dioxide assimilation. Now let's start with light reaction. Light reaction mainly occurs in the thylakoid membrane. So understand, in the thylakoid membrane, this photophosphorylation is done by photosystem. So what is photosystem is our next discussion. So understand in this thylakoid membrane, here so many thylakoids I am drawing. On the thylakoid, you can see there is photosystems like this. So one of the thylakoid membrane has been enlarged and drawn in this diagram. So hope you can see a lipid bilayer in this thylakoid membrane. On that only the photosystem is located. And this photosystem is mainly made up of two things. One is light harvesting complex or antenna molecule, and at the center there is a reaction center. Reaction center is chlorophyll A, that is the main pigment associated with photosynthesis. Surrounding that, you can see there is hundreds of other accessory pigments, chlorophyll B, C, carotene, xanthophyll, etc. attached to a protein. So hundreds of pigments and proteins surrounding that chlorophyll A is called light harvesting complex. And at the center there is a single molecule of chlorophyll A. So that chlorophyll A at the center and this light harvesting complex together, that is actually called as photosystem. So photosystem, a normal diagram, let me show. So if this is the thylakoid membrane, you might have learnt in the Cell the Basic Unit of Life chapter, thylakoid membrane is having a lipid bilayer. Or the membrane structure you might have learned, it is made up of a lipid bilayer. Hope you know it's having a head region and a tail region, and there is a photosystem. So like this, a photosystem will be coming. So what is this photosystem having? At the center of the photosystem, they will be having chlorophyll A. A single chlorophyll A molecule will be present. This is actually called as reaction center. So it's actually called as reaction center. Surrounding this reaction center, there are many pigments that are coming. And it is attached to the proteins. These hundreds of pigments and proteins that are attaching. So there are hundreds of pigments and there are also some proteins that are coming here. These hundreds of pigments and proteins together is actually called light harvesting complex, LHC. So this light harvesting complex and the reaction center chlorophyll A together is called photosystem. There are two photosystems. According to the discovery, it has been divided into photosystem 1 and photosystem 2. In photosystem 1, chlorophyll A, maximum light they are absorbing is 700 nanometer. So the reaction center of the photosystem 1 is photosystem 1, the reaction center will be having P700. So photosystem 1, the reaction center chlorophyll A, maximum wavelength of light they can absorb is 700 nanometer. That is the far red light. And photosystem 2, the maximum wavelength of light that they can uptake is 680 nanometer. That is P680. So there are two types of photosystem. So what is photosystem? Hope you understood. Where is the photosynthesis taking place also? Hope you got an idea. So photosynthesis is mainly confined to green part of the plant. In the green part, you can see it is mainly confined to the leaf. In the leaf, mesophyll. In the mesophyll, chloroplast. In the chloroplast, there is thylakoid. On the thylakoid membrane, there is a photosystem. And photosystem consists of two things. One is reaction center, that is chlorophyll A, and surrounding that is the light harvesting complex made up of hundreds of pigments and protein that will be performing the light reaction. So I'm going to start with light reaction, photophosphorylation, making of ATP and NADPH using the light energy. So here there are two types of photosystem. One is photosystem 1 where the reaction center maximum uptake 700 nanometer light. Other is photosystem 2, where you can see the reaction center maximum uptake 680 nanometer light. So we are going to start with photophosphorylation. How in the thylakoid they can generate the energies like ATP and NADPH. So this is a thylakoid that I have drawn separately, one single thylakoid, and this is the thylakoid membrane. So on the thylakoid membrane, so I'm not drawing the thylakoid membrane completely. So like this, a lipid bilayer is coming. On this thylakoid membrane only, there will be the reaction centers. So here there is photosystem 2. This is photosystem 2 and there is photosystem 1. So like that, I have drawn two photosystem that is going to perform light reaction. In the photosystem 2, what all things are happening? This you have to observe. So photophosphorylation, here understand this is the thylakoid membrane, and outside the thylakoid membrane this is stroma, and inside this is the thylakoid lumen. And understand in the photosystem 2 they will be having a reaction center with P680. That means chlorophyll A molecule that will maximum uptake 680 nanometer light. Now one by one steps I am going to tell. Hope you understood. There is a photosystem 2 and photosystem 1. Photosystem 1, they will be having the reaction center where maximum light uptaken by chlorophyll is 700 nanometer. So photosystem 2 it is 680 nanometer, photosystem 1 it is 700 nanometer. So there is a single thylakoid. So I have told the structure of chloroplast. In the chloroplast, the grana, the light reaction is taking place. In the grana, it's going to occur in the thylakoid. So a single thylakoid we have taken out and drawn here. So in the thylakoid membrane, you can see there is photosystem 2 and photosystem 1. So photosystem 2, the maximum wavelength of light that they are going to uptake is 680 nanometer. This is the first step. And after uptaking the 680 nanometer light, so surrounding them there are hundreds of pigments and proteins. That is the light harvesting complex. They also do the same. They also take the light energy and they funnel it into the reaction center. That is the chlorophyll A. So light harvesting complex, that is antenna molecule. It is made up of what? It is made up of hundreds of accessory pigments and the protein that is actually forming the light harvesting complex. So they also take the light energy. They give all this energy to the reaction center, that is chlorophyll A. Now from here you can see 2 electrons are emitted. So there is reduction of this chlorophyll A molecule. That means uptaking this light energy, they get excited and they release 2 electrons. So losing the 2 electrons, they are actually getting oxidized. So they are getting oxidized by releasing 2 electrons, and there is reduction of a primary electron acceptor. On accepting the electron, there is a primary electron acceptor that is coming here. They get reduced. Its name is pheophytin. So let me write here what is PP that represent pheophytin. Pheophytin is the primary electron acceptor. Now there is a vacancy of 2 electrons at the reaction center of photosystem 2. So how it should be replaced? There only the water is utilized. Here there is a water splitting complex that is coming here. So let me show that water splitting complex. So at this location there is a water splitting complex in photosystem 2. The water splitting complex will perform photolysis of water. This is the third step where you can see there is photolysis of water. Photolysis of water will be releasing electrons, oxygen and H+. From these 4 electrons, 2 will be replaced here. This is the fourth step. So hope you understood step by step I am drawing. You should watch it properly. So here the photosystem 2 was first uptaking 680 nanometer light. They got excited. Second step, they are releasing 2 electron that is accepted by the primary electron acceptor, that is pheophytin. And to replace that 2 electron, there is photolysis of water that is occurring in the water splitting complex, which is located on photosystem 2 facing towards the thylakoid lumen. These all are questions. Where is the water splitting complex located? The answer, it's located on photosystem 2. It's facing towards which side? You should understand it's facing towards the thylakoid lumen. Which all minerals are involved in photolysis of water? This is actually from mineral nutrition chapter. Now it is deleted as per our new syllabus. Anyway, you should remember the minerals also that is involved in photolysis of water. One is manganese and the other is chloride. These are the 2 elements that are associated with photolysis of water. Then if you want the reference, you have to go to mineral nutrition. Then, so hope you got the 4 step. First is the reaction center uptaking 680 nanometer light. Second step, they emitted 2 electron, that is uphill movement. Then to replace that 2 electron, there is photolysis of water, liberating oxygen, and electrons are given to this one, the PS2 reaction center. Now from this pheophytin, you can see it will be taken up by the next electron acceptor, that is plastoquinone. So here PQ that represent plastoquinone. So plastoquinone is a not only an electron carrier but also an H+ carrier. That is a line given in NCERT. Most of the students get confused with that. The line is like this. Primary electron acceptor passes its electron not to an electron carrier but to an H+ carrier. So understand, this plastoquinone was asked in the NEET exam and all previously. So previous year questions, when you are going through, you will find the questions from this area. So plastoquinone, it not only transfer the electron. Using that electron motive force, they will be pushing the H+ from stroma to lumen. So whenever electrons are passing through plastoquinone, so they will take this electron and they will be giving to the next electron carrier. The next electron carrier will be here, that is cytochrome B6F. Like that, next electron carrier will be coming. So by the time what they do is, they will be pushing H+ from stroma to lumen. So understand, H+ is actually pumped by, that means it is low concentration in the stroma. Already H+ concentration is high inside the lumen of thylakoid. So against the gradient, that is from low concentration to high concentration, using this electron motive force, they are pushing the H+ from stroma to lumen. So like that, you can see the H+ is moving from the stroma to lumen of thylakoid, and it is done by plastoquinone. So remember, plastoquinone is a H+ carrier, and they actually push the H+ by energy. They are pumping H+. Pumping means it should be from low to high. That is what you should remember. And next person to which these electrons are going is cytochrome B6F complex. And finally, the electron will be reaching plastocyanin. PC, what I am writing, represent plastocyanin. It contains copper and all. So to plastocyanin it will be moving. So let me give a good recap of the topic. Understand photosystem 2, they are uptaking 680 nanometer light. From here you can see 2 electrons are emitted and it is taken by the primary electron acceptor, that is pheophytin, PP I have written here. To replace the 2 electrons here, there is photolysis of water and the emitted electrons are actually used for replacement at this location, liberating oxygen and H+. Then second, from pheophytin the electron move to plastoquinone, then to cytochrome B6 and F, then to plastocyanin. While the electrons are moving through these complex, plastoquinone is not only an electron acceptor or electron carrier, it is an H+ carrier. It will be pushing the H+ from stroma to lumen. Now what is happening inside the lumen? In the lumen, H+ concentration is increasing. So understand, due to photolysis of water and due to pumping of H+ towards inside, H+ concentration inside the thylakoid lumen is increasing. Question, what happens to pH? pH is decreasing because H+ is increasing means it's becoming more and more acidic, or the pH is decreasing. So here there is a proton gradient that is building up. This proton gradient has to be broken. By breaking the gradient only, ATP is generated. So how the ATP is generated? So this H+ will be pumped outside. You may be knowing about the integral proteins in the thylakoid membrane. Some of the integral proteins make protein channels. So let me show one of them. The one which is making a protein channel like this. This is a transmembrane protein channel. That means they are connecting from the lumen to stroma outside. So it is a transmembrane protein channel. This transmembrane protein channel on the thylakoid membrane, this is actually called as CFO particle. It is called as CFO particle. It's not CF0, it is CFO itself. So this CFO particle is a transmembrane protein channel. Through that you can see H+ is moving from high concentration in the lumen to low concentration in the stroma to break the proton gradient that is building up inside the lumen of thylakoid. So inside the lumen of thylakoid, if this proton gradient is increasing, this can actually resulting in bursting, or the pH is becoming acidic and all. So it has to be broken. So to break the proton gradient only, they start moving the H+ from high to low concentration through a transmembrane protein channel. So this process is called as, you might have learned, if anything is moving from high concentration to low concentration through a protein channel or carrier protein, it is called facilitated diffusion. So H+ is moving by which process? Facilitated diffusion. So H+ will keep on moving by facilitated diffusion. Now you can also see they will be having one more particle, CF1. That CF1 particle is having an enzyme ATP synthase. So ATP synthase, when this H+ is moving through that, conformation means structure will change. When structure is changing of this enzyme, there is attachment of two particles at this location. One is ADP and P, and later they will get converted into ATP. So this was proposed by Peter Mitchell, where you should understand whenever H+ ions are moving from the thylakoid lumen to stroma through this transmembrane protein channel and the CF1 particle, the conformation of the CF1 particle will change, or the ATP synthase enzyme will change. And when it is changing, two substrate will be attaching to that. One is ADP and the other is P. They will get converted into ATP. So it is coupled with the synthesis of ATP. So this is how ATP is produced in the light reaction. So I said in light reaction there is direct generation of ATP and NADPH. So how it is produced only, I just explained. So this process is using sunlight, hence it is called photophosphorylation. Now, in the photophosphorylation, electrons emitted from this photosystem 2, first it is going uphill. Then you can see remaining movement is downhill up to plastocyanin. Simultaneously, when photosystem 2 was starting, one more will start, that is photosystem 1. Photosystem 1 will be uptaking 700 nanometer light and they will be releasing out 2 electrons. These 2 electrons are first accepted by iron sulfur. Then from here, these 2 electrons are downhill to the next one, that is ferridoxin. So all these are electron carriers only, ferridoxin. So let me write it as FD, that represent ferridoxin, FD. FD stands for ferridoxin. Then from the ferridoxin you can see the 2 electrons are going to one enzyme, ferridoxin NADP reductase. So FNR stands for ferridoxin NADP reductase. So what they perform is reduction. That means they will give these 2 electrons to NADP plus. And for that reduction process, they are also going to take H plus from stroma. So by using H plus from the stroma and electrons from the ferridoxin, they convert this NADP plus into NADPH. That is also in the stroma side. Understand? Light reaction, though it is taking place in the grana or thylakoid, both the energy currencies like ATP and NADPH, that is actually formed inside the stroma. Using that only in the stroma, dark reactions are going to take place. Now, there is an electron vacancy in PS1 that will be given by plastocyanin. So, movement of electrons, please understand, these electrons were actually moving from photosystem 2 to photosystem 1. And there is also movement of electron from photosystem 1 to this NADP plus. So, NADP plus was getting electrons from photosystem 1. And that lost electron in photosystem 1 is replaced by electrons from photosystem 2. So this overall movement, if you are taking, you can see it's forming an Z scheme. So this is actually the Z scheme. And this is also called non-cyclic photophosphorylation because emitted electrons are not returning back. Emitted electron from PS1 is going to NADP plus. Emitted electron from PS2 is going to PS1. Emitted electron from PS2 is replaced by water. So that's all about photophosphorylation. Hope you got it. So first think like this. Photosystem 1, 2 electrons are lost and going to NADP plus, it get converted into NADPH. Photosystem 2, 2 electrons are lost, it will come to photosystem 1 and replace the lost electron. Photosystem 2, the lost electrons are replaced by photolysis of water. So this process occurs simultaneously like this. Now question area, all these electron carrier, they are located towards which side and all, it can be asked. Where is the water splitting complex located? It's in PS2 facing towards thylakoid lumen. Where is the plastoquinone located? It's on the thylakoid membrane facing towards stroma side. Where is plastocyanin located? It's in the thylakoid membrane facing towards lumen side. Where is ferridoxin NADP reductase located? On the thylakoid membrane towards the stroma side. Where is the NADP plus located? It's on the stroma side attaching to the thylakoid membrane. Like that, plenty of question you can solve from this single diagram. So this diagram making like this capsule notes are very important in your NEET preparation, because a single diagram itself you can actually do around 40-50 questions. So in this diagram itself plenty of questions can be done. So ATP synthesis also, I think it's well clear from the diagram. So ATP is generated by breaking a proton gradient. This was actually proposed by Peter Mitchell. So how this H plus is increasing and how its gradient is broken down and how they generate ATP, all this you should understand from the diagram. So whenever electrons are moving from PS2 to PS1 via plastoquinone, this plastoquinone will keep on pushing the H plus from stroma to lumen. Not only that, because of photolysis of water also, H plus concentration is increasing, breaking this gradient through this CFO and CF1 particle and changing the conformation of ATP synthase so that ADP and P can attach and they can get converted into ATP. This is how ATP is actually getting generated. That's all about the photophosphorylation. What I discussed is non-cyclic photophosphorylation because emitted electrons are not returning back. Emitted electron from PS1, it was going to the NADP plus. Emitted electron from PS2 was going to PS1 and that was getting replaced by the photolysis of water. So that's all about light reaction non-cyclic photophosphorylation. Remaining we will discuss in the next session. Okay then, bye.